Hi, my name is Ken Lasseson. I am the person behind the Microbiome prescription website, and this is one of my continuing series of trying to explain what I'm doing, why I'm doing it, and hopefully give people better understanding and better tools to be able to help themselves with a variety of diagnoses and conditions. This particular lesson is looking at identifying bacteria just by patient symptoms. Symptoms are usually what you describe to the doctor. They're not diagnosis, they're the symptoms. And it turns out that the symptoms seem to be better at identifying bacteria shift than your official diagnosis. It is a next logical step. So let's go in and do a quick recap. My first part was describing myself. I'm basically a data scientist, a computer developer and architect for over 4 decades. And the reason I got interested in microbiome was because I ended up having recurrent chronic fatigue syndrome, ME, and I discovered that the microbiome model worked extremely well for explaining why certain treatments worked for me. And I had a eureka moment and realized that basically chronic fatigue syndrome seems to be a microbiome dysfunction. And that explanation actually explains almost all the test results, all the variety of symptoms, and the challenges and difficulties of treatment for it. The next lesson dealt with identifying bad apples and missing apples in the barrel, i.e. bacteria that should be there or bacteria which are not doing the right thing. Either we have too much of them or too little of them, or a combination of the two. There's a bit of a randomness challenge because I looked at it from the point of view of saying, oh, why don't we go and correct it to match the ideal microbiome? Turns out there's no such thing as the ideal microbiome. Turns out that the ideal ranges or the normal ranges given by labs to do the analysis is questionable, to be polite. So went in and went through that, showing why, who, etc., and where the logic breaks down and why it is very difficult, because there are many factors that have to be factored in to get a valid normal range, which is age, DNA, diet, and ethnic background, amount of exercise, latitude, distance away from the equator, are all factors are being demonstrated to affect the microbiome. So it's basically interesting and nice to do conceptually, but when you look at the hard numbers and the studies looking at it, it falls apart badly. Then the last one looked at using PubMed studies. PubMed studies are where somebody either got the idea or realized that microbiome is part of an illness, a diagnosis. So they got a bunch of people who have the condition, a bunch of controlled people, the test on the two, and said, Eureka, we found that such and such bacteria is higher in the control group than in those with conditions, or the reverse. So we have a bunch of studies there. The interesting thing is sometimes the studies contradict each other, and we end up realizing when we dig into the study that the studies often are disjoint. You may have one being done on a Chinese group of patients, another done on European patients. The Chinese patient has one major group of bacteria low. The European has the same group high with the same medical conditions compared to their controls. Things such as typical diet and DNA comes into play, which could account for two different results. Of course, the question is, for you who happens to be 1/3 African American, 1/3 Latino, and 1/3 Caucasian, where do you sit in there? Should it be higher? Should it be low? What type of thing should I do? We don't know. Nobody has done a study on your type of person. A lot of the studies are very white, male oriented, So the things such as even gender often are ignored in the microbiome. And there are very distinctive differences between a female microbiome and a male microbiome. So just we finished the last one of the introductory series where we are trying to explicitly identify a bacteria that may need to be modified. The next set after that, we actually don't look at a bacteria, we look at a function, as in we need to identify which group of bacteria produces, let's say, too much folate or too little of some other chemical in the body, or which group of bacteria produces too much of an enzyme or too little of an enzyme. So at that point in time, we may be actually looking at 10 or 15 bacteria in your microbiome sample and try taking them as a unit, because this unit happens to cause excessive production or insufficient production. So the next three ones after this deal with end products, enzymes, and cake molecule. Cake comes from the Kyoto beautiful database of genes, so that we go from the bacteria, species, and strains in your microbiome samples to the genes which are in those same strains. And from those genes, we know what they produce. And from what they produce, we can estimate how much they produce. And voila, we have a different perspective on what's happening inside the microbiome besides just counting how many of each type of bacteria. We have a functional analysis. What functions are being well served or underserved or excessively served? Once we go through all of those bits of joys, we go on to sorting our apples. That is our bacteria for what you want to increase, decrease, and how do we develop a recipe to address it. As you can see, we have a lot of different ways of looking at bacteria, putting them into clusters, and then we've got the problem of how to make a recipe out of 400 different ingredients that does or that produce a palatable or good product, which is the recipe issue, and we'll come to that in time. The other last thing is looking at future perspective. Right now, we can work only with data that's available, and future perspective, I'll talk about what I would like to see being researched and developed to make this whole process more successful. Okay, so let's go on. One of the interesting things, for me, it came out of ME/CFS, where the diagnosis has no lab test that shows you have the conditions or not. What it is, or at least at one time, it keeps changing, was that there will be 20 symptoms listed, of which you need to have at least 4 of those 20 symptoms. If you have less than 4, you no longer have the diagnosis. You could have 18 of the 20 in some cases. And the catch is the diagnosis is effectively the symptoms. When I was working through it, I realized that, and had to confirm when people started uploading samples and gave the symptoms, that the symptoms are reflections of specific mixtures or specific types of dysfunction of subsets of bacteria. In which case, if you have a diagnosis, that's something different. But if you're looking about symptoms, symptoms seems to be able to be tied back to specific bacteria dysfunctionalities. So, bottom line is bacteria patterns and symptoms seems to be far better associated than trying to do bacteria and a diagnosis. Often diagnosis includes a pretty broad paintbrush, and symptoms are far more detailed. Example is, if you look at something like hyperthyroidism, which I have at the bottom right, you see the symptoms, feeling tired, fatigue. You will see that with irritable bowel syndrome. You see that with chronic fatigue syndrome. You see that with a variety of other conditions. Gaining weight, you see that with a subset of chronic fatigue syndrome patients. Another subset has problems of constant weight loss and disappearing into almost nothing. Forgetfulness, brain fog, associated with a lot of things. For example, lactic acidosis will often have forgetfulness happening. Chronic fatigue syndrome will have it quite often. Fibromyalgia will have it sometimes. Irritable bowel syndrome will have it sometimes. Now, if you're looking at the diagnosis of one of those, and you get all those different mixtures of symptoms in your control, in the group you are doing analysis on, you will have, if bacteria associated with symptoms, you automatically have gotten a lot of noise, a lot of scattering of bacteria in your sample groups, because some will be high in things connected with feeling fatigue, others will be high in feeling connecting weight, and you can, in some cases, have so much noise happening, you don't get any results. Not because there isn't results, it's because how you are parsing apart the data happens to be at a very high level, so you just don't get the realization, oh, here is what is happening. It is this set of symptoms or this subset, which I never created. The subset of the diagnosis is too broad, and you have to subdivide it. In some cases, into 20 subdivisions. But then we usually hit a problem with medical studies that often can take 6 months to a year to get enough people with a general diagnosis to get a study group going. If suddenly you have to take a subset of that, it's not going to take a year to get, it may take 5 years or longer to get enough people to do the study, and that doesn't work very well when you're looking at trying to get grant money. So you end up trying to compromise how selective you are for your participants in the studies versus how long it takes to get them. And once you have that, you run and you hope to get results. So that's basically the challenge going back and forth. Now, the symptom approach has some differences from what you see in PubMed. PubMed study usually works on averages, and they usually assume a Gaussian distribution because that is how they calculate the statistical significance, something called the p-value, the probability value. That is, what's the probability that we would get the same thing happening by randomness. Most of the time, studies are less than 5% chance, that's 1 in 20, that it will happen by random. In some cases, 1%, which is 1 in 100, which sounds nice and sounds good. And if you're dealing with things like 5 or 6 specific lab results, then yes, you are pretty much in the home run. But if you're dealing with 600 different bacteria, 5% of 600 bacteria is 30 bacteria are likely to be significant by randomness. So that's one of the problems with the PubMed studies, which I talked about in my prior discussion. Symptoms, we use a different process. We do things in bins. So the rows, we have created a table. The rows are the symptoms, or with symptoms or without symptoms, effectively your control group and your group with the particular symptom or diagnosis. And now we divide it not by actual numeric value, but by percentile. So that in the first bin is everybody is in the 0 to 25 percentile range, 25 to 50, 50 to 75, 75 to 100 range. And now we find out how many is in each cell. If the condition and the control are identical, we have almost the same number in each one of the 4 bins. If they are not the same number, if one of the bins have a huge overabundance compared to the other or underabundance, then we can say, wait a minute, this is where something has happened. It could be right in the middle or it could be at the ends. And by doing this approach, we actually end up getting much better results than working with averages. One reason is we make no assumption as to what the distribution is. PubMed assumes a Gaussian distribution, or sometimes known as a bell curve distribution. And we can construct statistically situations where you get both rows having identical same average. No statistical significance using the PubMed approach, but with a specific range having an abnormally high, very statistically significant one. So, in other words, we are more sensitive to detect statistical relationships than using the classic PubMed average and standard deviation approach. Also, we have up above, you see 4 bins. We can actually do 8 bins, 16 bins. We can do as many bins as we want. But as we add more bins, the ability to detect actually decreases once we reach a threshold, because we're dependent on having usually at least 5 expectation of having 5 elements or 5 patients in each bin, preferably 10 patients in each bin to be your expected numbers. And then with that, we can actually get some pretty good statistics done. So I include a link to example of a page where you can go and play around with this type of bin. It's a Fisher 2x4 page to allow you to do online calculations. So you can go in, play around, and get a hands-on feel for it. Now, the purpose of this thing is to hint at what bacteria could be involved. Again, we're working on association. We're dealing with probability. We not are doing cause and effect. We are simply saying we have this relationship, and we think that the relationship is some ways causal. Experience from people I've worked with is that it is causal. You reduce a bacteria which is associated with a symptom. The symptom becomes milder, or in some cases totally disappear. People have told that to me when they have used the microbiome prescription site. Personally, I've experienced it using the microbiome prescription site. I'm absolutely free of all of the symptoms commonly associated with chronic fatigue syndrome, although one time I was sitting about 12 or 14 of them. None of them are there anymore. No fatigue, no brain fog, as probably witnessed by what I'm doing here. Able to concentrate well. We do on microbiome prescription, we do some shortcuts, mainly because computer time costs money, and my training is in operations research, and one of the things you learn in operations research is that a 95% of the way to the solution is cost effective. The last 5% may cost you 10 times more than what it did to get to 95%. Sometimes we refer to as a Pareto, P-A-R-E-T-O effect. So what I do is, okay, I use what I was trained. I do a few shortcuts here and there to save on computing time and to get results out faster and quicker. The results are continuously being published and updated and recalculated. Sometimes on some data are recalculated every day. So you literally go in and look at table one day and come back the next day, and you will see a slightly different numbers. They won't be identically the same because somebody has uploaded some more samples, put in more symptoms with the samples, and everything gets recalculated. So it's a living research site, not a static published paper. Again, I never change from traditional PubStead, which is done, published, cut in stone, move on. This is dynamically going. What does happen is our p-values, our probability values are much better. That is technically lower, smaller values. If you look down here, you notice we have e to the minus 6, minus 19, which means that it's not, there would be something like 18, you move the decimal point over 19 times to the right. Instead of 0.01, you're talking about 0.00000000003 chance of just happening by randomness. In other words, we have much stronger association, much greater statistical significance than what you find coming out of PubMed studies or averages. We also predict, and what we do is we look at the bacteria, look at all of the patterns we have identified, and then we see which patterns matches. What we usually find is that the best results are not at a single symptom, but symptom pairs. Sometimes 3 or 4, but generally symptom pair is what our amount of data works best with. We don't quite have enough to go up to using triplets. We probably will in the next year or two, but at the moment it's pairs. And what we do is we see if there's a matchup, and we look only at the strongest relationship, and we see 4 patterns here, and the sample which is uploaded has all 4 of those matches. So it implies to us there's a good chance that this combination of IBS and temperature fluctuations throughout the day is likely happening to the person who submitted the microbiome. We know nothing of the medical history. This is an attempt to predict from the bacteria what to expect. The surprising or good news, depending on your perspective, is that from the feedback I've gotten from people, is we seem to be sitting 80% accurate in these forecasts or better. Occasionally, I've gotten a very amusing email saying, "Excuse me, you were dead on, and one of the things which you predict I had is something I have, but I never mentioned to the doctor. I forgot all about it because I've gotten so accustomed to living with it that it has dropped off my radar." Well, you can't hide from your bacteria. Okay, let's go on. And what we have done is, for samples, once you have uploaded the samples, we actually try speeding up the ability for you to identify the bacteria by having a 2-step. We have discovered that there are pairs of symptoms that are statistically significant. So what we have done is we have created a drop down. So you select the first item, then you select the second item, and you can get the results together. Here is an example which I looked at one without the second item. You will get the result showing there. Over here, another one, and you get a result there. 2 different lists. We have, I think, we have bifidobacteria in common, lactobacillus in common between these 2. This one isn't on the bottom one. But when we put the 2 symptoms together, we get bifidobacteria is still there. We get Akkermansia, which is on this list. And we have somebody new showing up, which seems to be a unique bacteria whose presence or absence, in this case high values, is associated with this particular pair of symptoms. This is sometimes why it's interesting working from pairs, because you end up being more precise for which bacteria are actively involved, rather than dealing with a rather long list of possible bacteria. We actually want to reduce the bacteria down to the fewest numbers practical or pragmatically, because it means adjusting them becomes much simpler than when we are trying to do everything at one time, because affecting one bacteria affects other bacteria, and you end up with this horrible balancing act because things interact constantly. So, to do a quick summary, the symptoms the bacteria pick is the page loads, you set your samples, you have many samples, you can pick different samples, you select your first symptom. For example, I put in my age, 60 to 70, you'll get a list of everything there. You can go in and then put in second things like a brain fog, and you can go down there. And what you will find is that the values are significant are marked by little exploding bombs. It says on the right if it's a match or not, gives your count, then gives the range of values for the buckets. Some of the buckets are small, like here. This bucket is only a range of 115. This bucket is a range of 2400. This bucket is a range of 24,000. The ranges are determined by the distribution of the percentile. It's a slightly different way of looking at the data. Rather than trying to fix everything into fixed ranges, we actually using percentiles to determine the range of values, which seems to work very nicely so far in everything I've looked at. I've been quite pleased with the results. Now, at this point in time, I should do a quick reminder of when you look at the values, sometimes you see no value, a blank there. It could mean that you have 0, or could mean something else. It could mean the lab did not report it. For example, this particular bacteria, you see 3 of the 5 labs listed reported almost all the time. 1 never reported. 1 reported just 1% of the time. So if you have no value and you happen to come from bottom side or American gut, chances are toss away that information because that bacteria was likely not measured. So the 0 value means nothing. It's like saying, how many octopus do you have in your swimming pool, which you're going to build tomorrow? You have no idea if there's going to be any there because it's not measured, it hasn't happened. Another reason to ignore it is if it's not seen very often in samples. Here we have nice high number, 97, 95. Now, if the samples you have occurs just 5% of the time, that means that the significance is not as clear that it is significant. It is fuzzy. It could be just noise. It could be any one of 100 other things. So it's less reliable. It's less safe to include it. You're welcome to include it, but it should be done with a bit of caution. If you want to determine this, again, it's easy. Just click on the bacteria name and it will take you to the page that will give you all of this information. If it's a low value, it's rare, and small p-value, then it becomes really fuzzy. Remember the p-value, usually it's with some long number with an E, with a number there. If there's no E, it is definitely a low value and probably can be ignored. The bigger the E value, the more significant it is. But if it's rare, it becomes hard to say, is this just noise or is this a significant factor contributing? We don't know using this type of analysis. Another type of analysis, we may be able to answer that question. High value, usually assume that the bigger the volume, the bigger the count, the bigger the impact. Simple as that. So if you're matching a high value and you have a large number of numbers, high number of this particular bacteria, then the higher the number, probably the more significant it is for whatever your problems are. Now, if you click on the bacteria, you get a reference page that shows things such as what I just showed, things such as the percentage from different type of bacteria. But it will also, if you scroll down the page, will show you the symptoms that are most often associated with this bacteria, and they're generally done as pairs, and it will give what direction it is. So just generally seeing if you have low values, there'll be some which will be seeing high values, and then the probability of statistical significance of it. Now, if you do one step further and you go and click on the label here, it will take you over to a page where we'll give you the breakdown of what bacteria, the rank that is. Are we talking street level, county level, state level, and what type of relationship it is. First of all, what type of shift it is, which one, in this case, of the 4 buckets it falls into, and how strong the relationship, strong or weak, etc. Generally, we go for strong when we do predictions, or very, very strong. Otherwise, we give it for informational purposes. Some cases, if nothing comes out strong, you may want to consider some of the weak relationship. But here's where the sweetness comes. If you click on the values here, the page will explode out and you will see the numbers. If you take a look here, we have 30, 63, 74 numbers here. 74 divided by 4 ends up somewhere 80 divided by 4, about 19 each. So every one of these should have 19 in them. This one is close to it. We have a big bunch of numbers sitting here at the very bottom. The numbers aren't there. This one is sort of so-so. But what it means is that we find associations that are low numbers and this particular bacteria and the condition we're looking at appears to be unusual, appears to be statistically significant. Here's another case. In this case, we have the medium high. If we go up a level, break it into more parts, you see, instead of the expected 37, 44, 46, about 50 divided by 8, we are expecting about 6 per cell, and we find a big whopping bunch between 63 and 75 percentile, where we have far more there. And if you take a look, the ones beside it are high, and as you go further out, the number starts dropping off. So we sort of have a bell curve sitting around the 63rd to 75th percentile. Not bell curve on the count, but bell curve on the percentile. And now from this, we can go in and we can look back to see what the actual numeric values are and work from there. So we have a lot of numbers floating around. The main thing which we do, which is very critical, is we work from percentiles, not from raw numbers. And the percentiles, in some ways, maps to what the type of microbiome variations that happens naturally without forcing things into a bell curve. Now, symptom list keeps growing. I think I have something like 450 different symptoms now. And including the symptoms, we do include some diagnosis. And in fact, I need to revise to make sure and have caught up, is that everything which we have PubMed studies on, we also have the same diagnosis listed as a symptom. So if you have this as a symptom, you can put it in so that we have a second view of the same medical diagnosis, what PubMed articles have stated, and what we discovered with an alternative way of taking part in the numbers and using citizen science methodologies. In general, we need at least 10 samples to get any results. And as I mentioned before, when we're doing a chi-square, we really want to have 5 or 6 in each one of the cell, which means we really are looking for 20. 10 is iffy, 20 starts becoming reasonable. 10, I end up having to do some slightly different types of statistics to compensate. So ideally, we'd like to have 20 samples with particular symptoms in order to get significance. The flip side of having already 450 symptoms is that you can keep saying, oh, how about this symptom I have, or that symptom I have. In general, we don't want to go down adding in every symptom anybody comes up with or describes. Some cases, they will use one name for a symptom and somebody else refers to it by a different name. So we have information being lost by having 2 different classifications which may not match up. Also, we don't want to get a checklist of 8,000 or more things. Reason is the person who comes in to mark the symptoms going to be exhausted. They're not going to finish doing the job. If they don't finish doing the job, we have incomplete data and we have less information coming back to you as to what is going on. So the result is symptoms are clustered into groups. Some of these groups I'm probably going to do some revisions on. But basically, if you click on one of the buttons, they expand out and you get the symptoms involved in it. And you can just go and click the ones you have for the samples. Once you have clicked them, you just do a click and update. And that information gets saved in the database. And sometimes the next 24 to 48 hours, the database will use that new information and recalculate everything. Oh, everything. I mean, it's a good long job every day. So, key aspect is this is a promising method for identifying bacteria. We have a very strong association to the bacteria, much stronger than what seems to be coming out of diagnosis. But this method also allows us to test diagnosis, although the diagnosis is self-reporting. It's not a doctor applying criteria. Self-reporting can be problematic because people often self-diagnose incorrectly, but that's simply the reality which we try coping with. In some ways, it's not that bad because if the criteria for the self-diagnosis is things like internet, talking back and forth, it may be incorrect diagnosis in terms of medical, but generally the diagnosis would be consistent with the set of symptoms that the internet lore has associated with the diagnosis. So there is a bit of, we may not be medically correct, but the data was probably still going to be useful, and it seems to be. This is a totally citizen science. We're not depending on funding from an institution or grants to do it. We don't depend on my footing the bill, but I'm doing that now and asking for donations if you may have spare cash. If you don't have spare cash, forget about it. I can afford to do the funding myself out of the pocket. It depends on people annotating the symptoms, not only paying for getting the fried biome site, Nirvana biome tests done and uploading them, but they also have to go in and add their symptoms, which sometimes can be a bit frustrating because some of these sites will ask for your symptoms, and the symptoms they ask for often are as few. Sometimes they're asked in an awkward way. I know I've gone through a few of the sites and started to click symptoms, and I got fed up after symptom number 20 and just said, forget it, and left it in. So they left it unfinished so they get incomplete data, which means they can't do anything. Here, please, because you can see that it has rewards right now, get those symptoms done. So it's less speculative that, we may be able to use it. We are using it right now. So those symptoms on this side have a high value and have payback within 48 hours because those information will get recalculated within 48 hours. The symptom to bacteria is a very easy to understand concept, and that makes it easy for people to accept and to run with. We do have to be careful not to go and have excessive expectations. For example, keep subdividing into small interval. Once we hit a certain degree of the expected numbers in each cell, each one of our table gets below 5, we are in iffy territory. We really want to have the numbers higher, and if the numbers are higher, we have greater confidence that we are actually determining real relationships and not just the bad roll of the dice. What can sometimes be very illuminating is sometimes we find comorbid conditions may have 70 or 80% of the same bacteria shifts being reported. The difference is why somebody has one set of symptoms and somebody else has never set of symptoms, and more people have both sets of symptoms because the bacteria are overlapping and it's only one or two additional bacteria that seems to be the difference between somebody suffering from, let's say, gluten sensitivity to multiple chemical sensitivity to light sensitivity, all of which may have only a few bacteria difference between them. And it helps understand why we have comorbidity. Okay, so I have gabbed on and on about it, so let's go over to the site and see things in action. Hi, so here we are on the site. Give me 1 second to move myself. The first thing is to log on. I'm supposed to would have received an email with a click to immediately log on to view your data. If you don't have sample uploaded and just want to get familiar with what's going on, Biome site has contributed one of their data sets for us to use for demo purposes. Click there. If you want to see what they offer their site, just click here. We have discount codes and on there, and actually should add the discount codes to this page for people who are interested, as well as on the upload page here, which shows the type of data you can expect coming back from different people. I do have a bunch of discount codes, and should make sure I have a link to them there. Okay, let's continue onwards. Biome site there, and we are looking in the world of symptoms. So suppose you just finished uploading a sample. Probably the first thing you want to do before you do anything is amuse yourself and see what the predicted symptoms are. So here we have predicted symptoms. You see here it's 11 and it's rating 1 of 10. Simple drop down so you can see all of the symptoms, and all the symptoms are listed there. The number of matches which we find have a strong relationships are here, and the number of matches to pathogens here. Sometimes if you go in here and if you go and click on one of these, broken page. Sorry about that. I'll get that page fixed in the next day or two. There are a lot of pages and a lot of links, and occasionally things go wrong. So what we have here is we have that. Now up to the top you have the significance threshold. So if something doesn't meet at least 0.95 in terms of the number of matches, in other words, if we have 95 100 things to match, we need to get at least 95 matches there. If we drop to something like 0.8 and do an update, we no longer have 11, we have 29 matches. And you can see some of the matches are of interest. Here we have 9 patterns and we match 6. The predictor gives a 0.9. There's a bit of fancy math behind that, but basically it is a indicator for how close in terms of a fuzzy cloud the things are matching. It's data science, machine learning. Let's skip the technical details. So we can adjust that to get more or less. The default value is very high. As you drop the value, the reliability of the prediction goes down. You could find that you can knock the number down very low and still get correct predictions or high percentage of correct predictions. Okay, now once you've seen the magic of the symptoms, you probably want to go in and enter your own symptoms. The symptoms when the sample was taken. If you can remember that, hopefully you did. Make a note of the symptoms there, as many as you could. And now we just go in. Typically what we do is we pick age, and mark what the age is. And actually for here, I'm going to correct the age to mine. If you click it again, it goes and hides the value. Over here we have gender. Well, perhaps I should be change my gender. You can put in bogus data or bad data. Please don't. Try keeping the data as accurate as possible. Although with statistics it will hide bogus data to some degree, but too much bogus data becomes more and more problematic and counterproductive to most people using the site. So we have things like neurocognitive, brain fog, absent-mindedness, monofocus, slowness of thought, unable to focus, vision or attention. Again, mark the ones which are appropriate, collapse them on afterwards. By doing the expanding, collapsing, I have found most people are less overwhelmed by the list of symptoms, because if I expand all of them, it gets to be a very, very long page. If you are actually interested in doing it offline rather than online, what you can do is go over to here to look up symptoms reported, and it will list all the symptoms by their grouping and the names, and it will go and show you how many people have reported each one of the symptoms. So it will give you a perspective, and you may want to print that off and go through and highlight the symptoms you are on paper. Some people find that to be a much easier way of doing it than on or off. Or if you have cognitive impairment, doing it on paper is much easier, and then you could get a friend to enter it into the page. So you have put in your symptoms, and probably the next thing you want to go is over to bacteria symptom summary. Bacteria to symptom is the same, except it does live real-time calculation, which means it can take up to 5 minutes to get a page done. Most people do not like that type of turnaround. We have to be snappy. I can't afford the massive machines to have a snappy page doing this type of calculation. But what I can do and have done is build a database of pre-calculated numbers which are periodically recalculated. And so what we have here is your sample at the top, and you have your double set of symptom picks. So you can go in and do something like, okay, I'm going to pick my age, and it will now come and show what, for that particular age group compared to all the other age group, what type of shifts are seeing. Some of these you'll find are in PubMed literature in studies on age, and finding that, guess what, they match up. But apart from that, you will find that the shifts are here. For example, certain type of bacteria tends to be higher in this age group, other happens to be lower. The 0 to 16 age group has a very different profile than adults and the quote elderly, like I'm probably becoming. And now you can go on and take a look at the second one. The second list is filtered by the first list, so the only thing on the second list are things where there are results that are statistically significant, and it has the first list. For example, you could do irregular heartbeats. And here it has it, and guess what? Almost all of the irregular heartbeats are connected with low amounts of a whole bunch of bacteria. And I see that, oops, I have, or correction, this is on my sample. Thank you. I was wondering there. But this person's sample, if the person happened to be in a 60 to 70 age range, may well suffer from irregular heartbeats. I could go over and check something else, like it's just person A type, and a person with blood A type, we have certain types of bacteria which are higher and certain types which are lower. So not only do we have age, gender, diet, DNA, we also have blood type coming into, but then again, blood type and DNA are associated. So it's not surprising, but it's nice seeing that we actually are seeing associations of certain types of bacteria with certain blood types, which of course makes things even more complicated to know what exactly is going on. But that is the recipe, and we'll come to that, opening that cookbook and learning how to cook a couple of lessons down the line. So that's basically it. Now, if we go over and click on any of these, okay, I'm going to go and the highest, okay, which ones has a high probability? Okay, let's go and I'll click on this one. This one I have a low count, and let's go over and take a peek. Just comes from Microbiome. We see that 96% of samples have it, so it is common. I can come down here, I can take a look, and here's where I'm lying. And in general for these charts, there's a long, flat, straight area when you're doing a log value. Happens to be how the microbiome seems to work. So anything on the line up to about this point here would be normal for just for the high point, and for the low point I would actually put the point here, because this is when the line suddenly changed, and that I'm starting to realize means that's outside the normal range. In this case, 40% of people are outside of what appears to be a normal range. These people have a very sudden dramatic drop in the values, and here's this sample sitting down here. Although it's a 20th percentile, it's on this ugly downslope. I could go over and say, okay, let's see this by percentage. And oh, I can say why. I have practically 0. So the normal range we saw up above is somewhere between 50 and, probably somewhere between perhaps 2.5% up to 5.5% would be a normal range in terms of percentages. But if you look at the log value, it makes the range that much clearer. It's the area where we have the big flat line in the middle, and the moment line starts going up or starts going down, you are, in my humble opinion, outside the normal range. And that's mainly because it indicates that the underlying behavior is altered for those ones. Okay, now let's continue on down to this page. We have from citizen science, we have all sorts of things. And guess what? You see variety of combination, and we see old type showing up constantly down here. Direction is low value there, and I picked old type, and indeed go there. But we also see other things such as old type is common, but old type has problems remembering things, inability. So in other words, we have the old type and certain types of cognitive issues are appearing to be as a pair. If I click one of these, we'll go over to the fun page, and the page I broke, I forgot the S in the link, which is why the page broke, so it's an easy fix. And hopefully we'll be there shortly. And we go over to the page. Actually, the page we go over is the one that does a recalculation. In other words, you're going over there based on pre-calculated numbers. Now you're going to look at the numbers which have been recalculated, so you have the absolute latest numbers. And down here you will see a variety of bacteria, what rank they are. Remember, rank is like street, neighborhood, borough, city, state, country. So you'll quickly learn things such as a genus, it includes species, etc. And we'll see some things having high and some things strong. And we see here, because we have a large number of people with just set of characteristics, or with just particular type of bacteria or characteristic, we are able to see multiple breakdown. Again, if I click on one of these, like there, what we see is we see what the breakdown is, and we notice that there is an absence in the middle and a spike at the high end, also almost just a spike at the low end. We only identify one for the purposes here, but often going in and taking a look at it, you will get some bit of insight. So we have high and low here. If we go and click on strong here, we do further expansion, and what we now see is the 88 to 100% is where it's really high. Everybody else is much lower, and the lower number sort of starts just the 40 down the bottom isn't as acute. And then if you go once more, we see that we have a whopping big at 26, and all the other numbers are very small. So it means you get to go and see where in the bins, in this case in 16 bins, if we break the data apart, we get a walloping big in one particular range, which is nice. So now you know what range. This is percentile range, remember? So you have to go back to the charts right, in order to find out what value range that is. But it will identify where the relationship is happening, and it gives you greater confidence, and you can see the numbers yourself. And I'm finding with bins, they're much easier for people to understand what is going on. So that's there. Okay, now let's hop back. So that's from citizen science, and all those are listed there. This table is nicely sortable, so you can click the p-value here, so you can have the one which has the strongest relationship first, and then go down from there. And we have 100 relationships with some statistical significance. So we have all sorts of things interested connected with this particular bacteria. And we see also what it produces. It produces two things. And we see how often it's found in samples. Remember, we need to actually go up to here to see the relative report, and particularly for the company you're using, how often it reports, because that's important. Because again, some companies are better, or the software is biased to reporting certain bacteria. Sometimes they see the same raw data, but their software will interpret it a slightly different way. Long story there's a recent PubMed article about take one lab sample, send to 72 labs, you get 72 different results, although the input was identical, because there's no standardization. There is no standardization, which means we have a problem, because PubMed articles may be done with one lab, another lab to get different results, may be done with a different lab, different physical equipment, different algorithms. So in one sense, they are incompatible. And what we do by aggregating stuff is we try doing fuzzy logic. Okay, so we have there, and now we have medical conditions. If they have been reported for any medical condition, for example, low levels are associated with psoriasis, high levels with Sjögren's or dry eye syndrome. And then down below, we have a lovely bunch of information about what foods or other things increases or decreases this particular bacteria, which just comes back to the recipe cookbook, which we'll be coming to later. So that gives you information about a particular bacteria. And I think basically at this point in time, I've got only one thing left to do. I'm going to, I have a match here and values, and where was the one I was looking at? This one here, I have a match with values. So I'm going to take those two bacteria, and I'm going to click this button, and it will add it to a hand-picked selection, which means I can be absolutely specific when I start working on recipes, which bacteria are to be included in the recipe. I click that, goes back to the page, and we have hand-picked bacteria here. And we can now look at the hand-picked taxa selections here. Oops, sorry, that's a cookbook. Let's get back, and we'll come back to the cookbook later, but right now we're just going to look at what bacteria are included. And these are ones which I have selected here, or for other things, to do a hand-picked bacteria. Usually when you are doing it, and I'll review this when we go to the cookbook, you want to have probably 10 or less bacteria if you want to have the best results. More bacteria you have, the more complex the recipe becomes, and then you have the problem of contradictory results. For example, barley may increase 3 of these and decrease 1 of them, which is ideal. So you look for something else which will work well for all 4 of them, and you find that suddenly restricts you to very few, if any, substances that magically works for everything. So we have to do compromise when we do recipe. But I think that covers most of the symptom aspect. Let me just click on here. Okay, so that's it. Using symptoms gives some pretty powerful information to be able to detect bacteria. And it's all coming from you. You are the contributor of both the microbiome samples you have gotten back from one or another direct retail labs, and you have contributed your symptoms, and as the net result, you will probably win and gain information, and other people will. So it's a cooperative game with high success, and more importantly, I've tried making sure that all the data is open and easy to view, and hopefully reasonably easy to understand. Sometimes when you're dealing with statistics, things become complicated or obtuse. So I do the best I can to make it intelligible. Okay, have a good one. And next time we'll go on to look at functions from the bacteria, groupings rather than individual bacteria.